Hi, good morning, participants who are here. Welcome again to the breakfast, UMHealth at breakfast. Breakfast at UMHealth. So today we have Dr. Cheong Fei Wen. So let me briefly introduce you to Dr. Cheong Fei Wen before she begin her talk on CRISPR-Cas9 related studies that she has been doing for the past years. So Dr. Cheong Fei Wen obtained her bachelor's degree in biomedical science with distinction from University Malaya. She passed her PhD with distinction and is currently holding a post of senior lecturer in the Department of Parasitology, Faculty of Medicine, University Malaya. So since PhD, she explored into several malaria-related aspects, including protein expression of Plasmodium knowlesi, merozoite surface proteins, immunogenicity study using animal model and epitope mapping. Her current research interests include phenotypic and genotypic measures on Plasmodium species resistance against antimalarials, studies on antimalarial properties of natural plants, immunogenicity profiling of potential malaria vaccine candidates, and genome engineering in Plasmodium using CRISPR-Cas9 system. With a deep passion for research, she has been awarded to attend several international workshops, including Unku Omar Newton Fund Researcher Links Workshop, Neglected Disease in Southeast Asia, Building Capacity in Epidemiological Modeling by with Imperial College London, UK, and also Malaria Experimental Genetics Workshop by Wellcome Genome Campus Advanced Course, UK. She has also successfully obtained and participated in 13 research grants with a total amount exceeding RM 2.1 million. With that, she has published about 30 ISI indexed articles and serves as an editor for several journals. On the other hand, she has assisted the department to develop a new master by coursework program, Master of Medical Parasitology and Entomology, and is currently the program coordinator. With her interest in academic curriculum, she has also been appointed as Program Quality Assurance Expert of Faculty of Medicine in 2021 and Program Quality Assurance Expert at university level in 2024 to assist in program curriculum related methods. In 2025, she has taken up the role as Faculty of Medicine Deputy Quality Manager, Pro-K, and also stakeholders feedback. So without further ado, Dr. Cheong Fei Wen, the screen is yours.
Without further ado, I'll start the presentation. So thank you, Dr. Arun, for the introduction. To help CRISPR-Cas9 in genome editing for malaria research, that we briefly go through the principles of and also what I before we it's just it's yeah it's a disease like meal and when they are taking the blood meal. So in a global scale, there are 200 happening worldwide, and when we look into the it's blocking. I think this is fine now. Okay. The quite successful in controlling the humans. Therefore, the research that in our research team very much on these species, which is Plasmodium knowlesi. And some brief idea about what is CRISPR-Cas9. This genome editing tool has been first described back in 2012 that it has high efficiency for genome editing. It can actually target multiple gene editing for simultaneously at the same time, and it can achieve a very precise repair. And we're going to talk about how that happened in the coming few slides. And this system is actually derived from the bacterial defense systems against virus. So what happened is when the bacteria is infected by the virus, they will start to collect the genetic materials and information from the virus. And then they will store this information in a cassette called CRISPR cassette. So in a nutshell, what CRISPR cassette work is, it works like a memory bank. And when the bacteria is next again infected by the virus, they will start to screen through this CRISPR-Cas9 to pull out relevant information, and then they will generate a short guide RNA and with sequence complementary to that. In this case, so they have generated this DNA which is complementary to the target. They will eventually use the nucleus called Cas9, which they will eventually make a double-stranded break in the virus and then disable them. So this is from the defense system of the bacteria. And researcher has found that this system is not only applicable to the bacteria, but it can actually happen technically in every cells as long as you give them the correct guide RNA targeting the right target and also the Cas9 protein. And what happened in general, after your DNA has had a double-stranded break, by nature, our cells will try to do the repair by themselves. So they are pathways that they used to happen randomly, that they will introduce insertion or deletion of the nucleotide, which is error-prone. And what we're trying to focus when we talk about CRISPR-Cas9 precise editing is homology-directed repair, or HDR. In this case, once you have generated the double-stranded break by the Cas9 protein, what you need to do is you can provide a DNA donor template that is carrying what specific change that you want. And if we can look into this schematic diagram, you will need to give them a template that they are actually homologous at 2 arms on the 5' and also at the 3', which they are homology to your target sequence. They are highly similar in the DNA sequence. And what you tend to bring in for the changes, you can put them in middle. So in this case, it will actually go through a double crossover that it will loop in the entire changes that you want into the genome. So therefore, it leads to the precise editing. And what CRISPR-Cas9 can actually work in Plasmodium, in fact, it's not just Plasmodium, but technically every cells, that first of all, if you want to study about certain genes that you have identified the target genes, but you want to know what is their function and what is their role. So what you can do is you can perform a knockout by disruption of the gene and see what's going to happen in their phenotypic characterization. And sometimes you will realize that if the genes that you're working is an essential gene, you wouldn't be able to generate a transgenic line, because whenever you try to knock out the gene, the parasite will not grow. The cell cycle will be arrested, so that you will realize that you have multiple attempts, but you still fail to generate the transgenic line. So what happens whenever you need to study the essential genes, you can actually incorporate a conditional knockdown system. In this case, for instance, you can use the destabilizer domain, or you can use the GLIM-S ribosome system, that you incorporated this conditional knockdown system in your design. So you can actually control when is the time that you want to knock down or silence your gene, or either up-regulated them or either down-regulating them, whenever that you put in an inducer or stabilizer molecule. So in this case, a researcher will be able to study the essential genes as well. And then in most of the current settings, what we are usually doing in our normal studies, we tend to perform forward genetic studies. What does that mean? Forward genetic studies is that we used to observe the phenotypic characterization. For instance, whether the parasite is resistant to the anti-malaria drug, whether the patient is having certain disease traits, and so on. And then we will try to link to the genetic level. What has gone wrong? What is the mutation that is happening in the parasites of the patient? So in that case, with the applications of CRISPR-Cas9, we can actually do the reverse way, by reverse genetic studies, that we have already pinpointed a certain mutations that we think it is actually linking to the phenotypic characterization. So what you can do now is you can incorporate these target repairs, whatever point mutations that you want, into the genome, and then only you evaluate the phenotypic characteristics. For instance, we have found out several markers that based on literature that are saying with this point mutation, the parasite is actually going to be resistant to the drug. is that the real scenario? What you can do is try to incorporate the point mutation into the parasite, and then you evaluate them phenotypically. Are they really resistant? So in this case, it's going to give you a strong association and linkage between the genotyping and phenotyping analysis. And also sometimes we also try to use a easier way in order for us to identify, detect, and also observe our parasites. So what we can do is we can just try to insert a tag, either immunotag or fluorescence tag, and we usually insert them in the 3' end, so that after the transfection, you will be able to detect or observe your parasite easy by using serological method, and also you can use microscopic examination for detecting the fluorescence, so that you can see the patterns and also expression of your protein at the time point that you want. And these are some of the examples that way earlier, once the system has been introduced, people have started to use that as in Plasmodium falciparum. For instance, like in this paper, they have actually generated the anti-malaria resistant parasite strain by introducing the point mutation. And recently, people also starting to use the conditional knockdown system to try to control the production of the gametocytes, which is the sexual stages in the Plasmodium. And in 2020, CRISPR-Cas9 genome editing has also been established in Plasmodium novocyte, which is the system and the species that we are using in our study. And now I would like to bring back your attention to Plasmodium, that the life cycle actually involves the host, which is human, and also the mosquito. And of course, Plasmodium novocyte, we involve the other host, which is the macaque. But in this case, our study, we want to focus on the erythrocytic cycle, which is what's happening in our red cells. And at the late stage of the parasite, we call it schizon. When the schizon starts to rupture, it will then release the parasite called merozoite back into the bloodstream. And this merozoite is going to invade into new red cells and then continue the life cycle and causing the clinical manifestation and pathogenesis on that. So what happened when the merozoite is trying to invade into the red cells is, first of all, they will actually try to attach to the erythrocytes membrane, and then they will reorientate themselves, and later on, they're going to form an irreversible tight junction formation, which means that there will be ligand from the parasites and also a receptors from the red cell. Then they will form a formation, and then eventually they will start to invade into the red cells by forming a parasitophorous vacuole that is completely sealing themselves away from the red blood cell cytoplasm. And after the successful emission, they will start to replicate and propagate again, and then this life cycle will then continue. And we have found out that in Plasmodium novocyte, we realized that Plasmodium novocyte Duffy binding protein alpha is one of the key ligands that it interacts with Duffy antigen receptor for chemokines, or in short, DAXI, on the erythrocytes red cell, erythrocytes membrane. So this interaction is actually facilitating the parasite to invade into the red cells. And over the years, our team has actually spent quite a number of efforts on trying to study on PKDPP alpha. And we realized that because the malaria cases and also severity has been reported rather higher in the Malaysian Borneo compared to Peninsular Malaysia. So therefore, we are actually in deep interest to see what actually leads to that discrepancies. And is that anything to do with the genes in parasite? So we have actually tried to study this by using the isolates collected from Peninsular Malaysia and Malaysia Borneo. And interesting enough that we found that the isolates collected from these two areas, they are actually exhibiting genetic diversity and leading to two distinct haplotype groups in these two areas. And further on, we also found out that the erythrocyte binding activity for the two haplotypes is actually different when they derive from different geographical area when we use the mammalian protein system to perform the experiment. And later on, we also raised the antibodies in animal models for these two haplotypes. And also we found that they exhibit different immunoprotectivity in vitro settings. But there are limitations happen in our current work, is that all the works that whenever that we need to use Plasmodium knowlosi in vitro culture, this culture is actually derived from the strain is derived from Peninsular Malaysia, which means that this culture, this particular strain is actually carrying the Peninsular Malaysia haplotype, especially for PKDPP alpha. And so far, there's no in vitro culture established for Malaysian Borneo till date. So what makes us think in our mind is that there are single nucleotide polymorphisms that is highlighted in blue, comparing the two haplotypes. So what makes us think of that? Would all these genetic polymorphisms in these haplotypes is going to affect the parasite invasion ability, in fact affecting their growth and also efficiency? So what we have planned out in the study is we have the wild type genome, which is carrying the Peninsular Malaysia haplotype. We are trying to replace them with the haplotype from Malaysia Borneo, which is the Sabah Sarawak strain. So ultimately, what we have done is we have tried to we have constructed the plasmid. First of all, of course, if we go back to CRISPR-Cas9, we will need to provide the system for a guide RNA and also the Cas9 so that they can target the correct position. And also at the same time, not to forget, we are giving them a donor template for repair, which is carrying the sequence of Malaysian Borneo haplotype. So after we have constructed the plasmid, we grow the parasites in vitro. And once we have sufficient amount on that, and with high parasitemia yield, we will concentrate the parasites, and then we will do a transfection using Lonza 4D nuclear factor. Actually, there are many systems can be used. It's just that we found that this system is rather efficient and give us a good result. So what we do is we just need to take a cuvette that is provided by the system. We put in the parasite and also we put in the plasmid together, put in the system. It's going to give you electric pulses for just like 2 seconds. That we will then further continue for post-transfection. After the transfection work, the parasite will actually, most of them will die off. So we will need to maintain them in the system, in the culture for several weeks to slowly wait them to come back and recover. So what happened in the system is we will also give them drug selection, which means that only those parasites is carrying the plasmid will be able to survive through the selection. And after that, we want whenever that we are working downstream, only single clone. What does that mean? Is that what we do is the parasite is 12 plate, assuming that there's only one parasite in every plate. And then we do certain selections in order for us to zoom down to select the clones. After that, we perform genotyping on the transgenic clones just to make sure that that parasite is really carrying what is the carrying the PKDP alpha Malaysia Borneo haplotype. For all the confirmations, we will then perform functional assays to compare between on their morphology, their invasion ability to the red cells. So this started back in 2024.
Yes, we can, Dr. Fei Wen.
Is this where I stopped previously?
You are now at preliminary findings 2024.
Yeah. Okay. So this project has actually started to be conducted in 2024, and now we have established the transgenic clones carrying PKTBP alpha Malaysian Borneo haplotype, and we after one round of observations in our experiment, we realized that there are apparently no trend for between the two. And also the C in the shadow for both lines. But interestingly, we noticed that one parasite in each red cell is higher in the wild type than the transgenic line. This experiment will need to be repeated to ensure the reproducibility of also reliability of findings. And what is the ongoing works for now? The antibodies from the two have. See now? I'm sorry, I think it's technical glitch.
Yeah, can.
Okay, so we are towards the end of the presentation anyway. So we will plan to evaluate the both the raised antibodies to see whether are they haplotype specific that or they generally can give a general protection across the different haplotype. And at the same time, we will also try to we will also try to perform a site-directed mutagenesis, which means that so far, based on the previous studies, there are currently no information about what is the crucial amino acid residues on PKDPP alpha per se. It is truly leading to the interaction and also the DAXI and also the invasion. So what we are going to do, we're going to form directed replacing the so-called predicted important amino acid residues with inert amino acid, and to see whether this will truly impair the invasions of the parasite into the red cells. And also, our team is also in progress of manipulating some of the gametocytes production related genes in order for us to have a stable and also high yield of gametocytes production, so that we can actually use this gametocyte to perform a transmission study in the mosquito vector, and therefore we can kind of like complete the life cycle in the mosquito laboratory settings. And since we have actually established this system in the department, that it actually can be applied to all kind of studies involving other parasites, organisms, and also diseases, as long as you have a good database to design the guide RNA to minimize the off-target issues, and then it will actually be applicable for all. And I think that's the end of my presentation. This project is actually funded by FRGS under MOHI, and I would like to take this opportunity to thank Prof. Lau, the head of Department of Parasitology and the main PI for the research team, Prof. Fong and Prof. Indra for always giving me continuous support. Mr. Rishi, very important person, that the PhD candidate who carry out all the works and the project is still ongoing. Dr. Pang, that is always giving us statistic analysis support, the research team members, and also the department colleagues and also students. And thank you for that, and I'm happy to open up for discussion.
Thank you, Dr. Fei Wen. That was a comprehensive and detailed presentation. So any question, or we have a question here already. So may I ask, the question is, may I ask clinically, what would be the endpoint and how would your work help treat or prevent this disease?
In this case, for the current setting in our expert, thank you for the questions. In this case, because we are trying to see the genetic polymorphism is actually leading towards a difference in terms of the protection. So by knowing the idea that whether this is important, and if we can identify the crucial residues on that, it's actually going to be helpful when we are doing like subunit vaccine designs, or even including like the raise of antibodies and also the therapeutic designs, that you can actually pinpoint those important point mutations that will need to be included in the designs. So for now, this work, we are actually still in the in vitro setting, but in terms to move into a clinical phases, that will require a longer and also more proper planning on that.
Okay, thank you. I hope that answers your question, Sarah Ahmad. Any other questions, any burning questions from the floor? All right, looks like there's no much questions here. All right, I think if there's no much question, maybe I'll pass it over to the next moderator to moderate the next session.
Yeah.
Thank you, Dr.
So
Cheong Fei Wen.
thank you. Just in case you have any questions, you may just look for me. We can have a further discussion on that. More towards like the general idea, also technical. Thank you very much.
Thank you, Dr. Cheong Fei Wen. So I'll pass it over to the next session. Prof. Ivy, the screen is yours. Thank you
so much.